Okay, y'all, this is the video for chapter 5 lecture. We're going to go to slide 20, and all of this is about cell membranes and mainly phospholipids, and just reminding you guys that the phospholipids are composed of a polar head group and the nonpolar fatty acid tails that create sort of a nonpolar barrier around each cell. Okay, so let's get into it. So membranes are composed of phospholipids, and the purpose of the cell membrane is to create that nonpolar barrier so the cell has control of which molecules or which ions come in and out of the cell. Because most of the molecules we're working with are polar, which means that they can't get through that nonpolar barrier, and so they need to go through a membrane protein to actually get through the cell. So we're going to talk about the structure of membranes, membrane fluidity, and then we're going to talk about some membrane proteins and the diversity of membrane proteins that exist. And then we will finish off with talking about pores, which are these sort of whole protein chambers in the cell membrane that allow stuff in and out. So the first, membrane structure. Membranes permit the passages of substances in and out of the cell because most of the things that are moving in and out of the cell are polar, and they can't get through the non-polar barrier of the cell membrane, and so they need to get through membrane proteins. So that membrane structures, they're a barrier, but they allow some stuff through. We have embedded proteins inserted throughout the lipid bilayer, and the cell membrane follows what's called the fluid mosaic model. So the fluid mosaic model states that all of the phospholipids, they're not attached to each other or to really anything. So they're kind of bouncing around, bumping into the phospholipids next to them, just constantly sort of like moving about each other. And the membrane proteins, if the membrane protein is not physically attached to the cytoskeleton or to the extracellular matrix, then those proteins are also fluid. They're also moving around. The only things that would not be moving around would be proteins, membrane proteins that are connected to the cytoskeleton or connected to the extracellular matrix. Anything unconnected, like the phospholipids or those membrane proteins, are just kind of like bouncing around each other, essentially. Okay, this is what a phospholipid looks like. It has 3 different diagrams. One is just the structure of the cell. Here's what it looks like in 3D, and then here's another model for it as well. This slide is to remind you that this is an amphoteric molecule, meaning that it has both a polar region and a nonpolar region within the same molecule. The polar region has a positively charged region and then a negatively charged region on that phosphate. Those are full negative and positive charges, which means that they're very capable of forming hydrogen bonds with water on the inside of the cell and water on the outside of the cell. Then we have fatty acids, which create the nonpolar barrier. The fatty acids can either be saturated, like what we see with this one right here. Saturated, remember, means that there are only single bonds between the carbon atoms, and so they form a straight line, it's linear. Whereas unsaturated fatty acids have at least one double bond between the carbons, and it introduces these kinks that actually makes the entire phospholipid take up more space. It increases the diameter if there are unsaturated fatty acids on the phospholipids. Okay, so here is a picture of a membrane and all of the different membrane proteins and glycoproteins, etc. So remember, our phospholipid bilayer is composed of phospholipids with the tails pointing inward and then the polar head groups pointing into the cell and out of the cell. We have proteins that are embedded within the membrane. In a second, we're going to talk about which particular amino acids need to be in certain locations in order to keep our protein embedded in the membrane. But membrane proteins can connect the cell membrane to the cytoskeleton. It can also connect the cell membrane to the extracellular matrix, Also, what's embedded on membranes is major histocompatibility proteins. We talked about major histocompatibility proteins on Tuesday's lecture. Remember, these are proteins that act like little flags, and every organism has a unique arrangement of different major histocompatibility proteins. So this is the way of the immune system recognizing self from non-self. Then we can also have proteins that are attached to the extracellular matrix, like collagen and elastin there. Also, membranes have cholesterol embedded actually within the membrane. Cholesterol is a nonpolar molecule, which means that it's able to be in this nonpolar region. What the cholesterol does is it helps maintain membrane fluidity. You want your membranes to be fluid. The phospholipids need to be able to move past each other, and the proteins need to be able to be fluid. There needs to be movement there for a healthy cell so that the cell can bring in stuff or have stuff leave. So what the cholesterol does is it introduces a little bit of space between the phospholipids, and the more space between them, the easier it is for them to maneuver about each other or past each other, essentially. Okay, membrane structure. So cellular membranes have 4 components. Obviously, the phospholipid bilayer, which is the main component, head groups on the outside, tails pointing towards each other. Then we have our transmembrane proteins that connect the cell membrane to the interior cytoskeleton network. Then we have interior protein networks as well, and cell surface identity markers that are part of the major histocompatibility complex, which allows the immune system to recognize that the cell next to me is part of the same organism. And then if a cell has different major histocompatibility proteins, then the immune system should kill that cell, because the immune system can safely assume that any cell that doesn't have the same major histocompatibility proteins is probably dangerous. In Tuesday's lecture, we talked about how these major histocompatibility proteins are really important for organ transplants. This is the major issue with organ rejection, is when a person gets an organ that has different major histocompatibility proteins than they have, and then their immune system recognizes that, and that can lead to organ rejection. But these are the common proteins that are embedded in the cell membrane. Okay. Let's talk about how scientists figured out the structure of membranes. It was partially done through this experiment called the freeze fracture experiment. Essentially what they did is they froze a tiny cell in like a tiny little ice cube, and then they used a knife to break open the cell. And what they did is, because right here, these phospholipids are attracted to the water via hydrogen bonding. And these phospholipids are attracted to the water inside the cell via hydrogen bonding. The fatty acid tails of our 2 layers of phospholipids are not physically connected at all. They're only put in that arrangement because water, due to hydrogen bonding, puts them in that formation. So essentially, it's a long-winded way of me saying that there's nothing holding together these 2 layers of phospholipids, nothing physically holding them together. So if you freeze them, the easiest point to break is going to be between our 2 layers. So they pull essentially one of the 2 bilayers off, and you can examine it under an electron microscope to look at which proteins are there or not. But this original experiment was used to show scientists how the phospholipid bilayer forms independently due to the arrangement of water molecules, and how if you freeze them, you can break it off at the easiest point to break, which is just between the 2 layers. So this experiment showed that nothing is physically holding our phospholipid bilayer together. They're just put in that formation due to the nature of hydrogen bonding. Okay, back to phospholipids. So just remember the structure of phospholipids. Phospholipid, the head group is extremely polar because it has a positive charge and a negative charge near each other, which makes that region extremely polar. And then we have our two fatty acid chains that create that non-polar barrier. The bilayer just spontaneously forms to get into this formation because water doesn't want to be anywhere near those fatty acids. Water can't form hydrogen bonds with fatty acids. Water wants to be near substances that it can hydrogen bond with, and it can't do that with the fatty acids. So it wants to get away from them. So it will position the fatty acids in a way where they're not touching water and they're pointing towards each other, essentially. Okay, this is what that non-polar barrier looks like. So we've got the polar head groups pointing into the cell and out of the cell, and then our non-polar fatty acid tails pointing towards each other. This is the non-polar barrier that we're talking about in cell membranes. Just that fatty acid layer, that is the barrier that prevents polar molecules or ions from being able to cross on their own. Okay, now we get to talk about the fluid mosaic model. So the fluid mosaic model states that the phospholipids and all of the membrane proteins are just constantly moving about and past each other. Scientists were able to show this with a pretty cool experiment where they fused together two cells. So one cell is a human cell with human membrane proteins embedded, and then we have a mouse cell with mouse membrane proteins embedded within the membrane. When we fuse those cells together, the phospholipids and our unbound membrane proteins are just constantly moving about each other, bumping, moving past each other. And then eventually over time, the proteins are all randomly mixed throughout the cell. So this experiment showed that the phospholipid bilayer is extremely dynamic. It's constantly moving and grooving, and any proteins which are not connected to anything, those are also moving and grooving, and they're moving around past each other, and eventually over enough time, they'll just be randomly dispersed throughout the membrane. Okay, now we're going to talk about environmental influences on fluidity. So we're talking about saturated versus unsaturated fatty acids. Saturated fatty acids form straight lines, whereas unsaturated fatty acids form these kinks. They go into space a little bit. So if a cell membrane was composed of mostly unsaturated fatty acids, if I'm a phospholipid right now, and I've got saturated phospholipids all around me, I can't move anywhere because I'm packed in so close to all of my other phospholipids. We're all packed in like sardines, and we can't move anywhere. So having a cell membrane composed of primarily saturated fatty acids leads to really low membrane fluidity, which is not good. So it's good to have some unsaturated fatty acids in the phospholipids. So unsaturated fatty acids introduce some kinks because of the double bonds, and they just make the phospholipid wider in diameter. All of the phospholipids wider in diameter, which just creates more space between the phospholipids. So now if I'm a phospholipid with unsaturated fatty acids, there's space. I might bump in, but there's space between me and my other phospholipids, so I can just move on past them, glide around, and everybody's moving past each other because there's actually room to move. So it is definitely a good thing to have some unsaturated fatty acids in the cell membrane so that the phospholipids and proteins can actually move past each other to increase fluidity. Other ways to increase fluidity is through cholesterol. So most cell membranes contain cholesterol, which can it actually helps just maintain a stable membrane fluidity. So at warm temperatures, if the cell gets too warm temperatures, molecules move faster. There is such a thing as being too fluid. So cholesterol in high temperature environments can actually make the phospholipids move closer together to decrease fluidity if the fluidity is too high due to temperature. Additionally, cholesterol can increase fluidity at low temperatures. So at low temperatures, molecules are moving slower, which would just naturally decrease fluidity. I'm acting like a phospholipid now with my slow movements. It would naturally just decrease fluidity. What cholesterol does is it introduces some space in between the phospholipid layers, and even if it's cold, it's introducing space for the molecules to sort of move past each other. The bacteria actually have a really cool way of dealing with membrane fluidity. They actually have enzymes that can change saturated fatty acids to unsaturated fatty acids and increase their fluidity at cold temperatures. Animal cells cannot do this, but bacteria, this is one way that bacteria can survive cold temperatures, is to just create some unsaturated fatty acids, introduce some space, and then they can maneuver past each other. Okay, now we're going to talk about membrane proteins. There are a bunch of different types of membrane proteins. We've got transporter proteins, which move stuff in and out of the cell. Enzymes are on the cell membrane sometimes. Cell surface receptors that might bind to a hormone and induce some sort of change within the cell. Cell surface identity markers, these are part of the major histocompatibility complex that we discussed. Cell-to-cell adhesion proteins, we talked about those at the end of chapter 4. And then last, cytoskeleton anchors, that are proteins that connect to the cytoskeleton inside the cell to keep the cell in a certain shape. If we look at these proteins, if I were to take off all of the words on this slide, you would probably still be able to figure out what these proteins do because of their shape. So first, this transporter protein is a protein that has a passageway through the protein. So what's happening here is they're creating a non-polar environment right here that allows polar molecules to cross through the non-polar environment by creating a polar passageway through the membrane. This is an enzyme embedded within the cell membrane. So this would be our reactant, which is also called the substrate, that would bind to the active site of our enzyme and induce a chemical reaction to make a reactant or a product. Cell surface receptors. So these have the ability to be bonded or bind with some sort of hormone or some sort of communication molecule to induce some sort of change within the cell. It's a way that cells can communicate with each other long distance. Cell surface identity markers. These are like little flags on the cell membrane that help the immune system recognize self from non-self. Next, we have cell-to-cell adhesion proteins. These are proteins that physically connect to one another to hold cells together to create tissues. You don't have a tissue if your cells aren't sticking together. So these are proteins that help stick our cells together, cell membranes together. And then last, we have cytoskeleton anchors, which connect the cell membrane directly to the cytoskeleton inside the cell. Okay, structure relates to function. If we go back to this slide, if I were to take off all of the words, like I mentioned earlier, you would probably still be able to figure out generally what these proteins are doing on the cell membrane. So all transporter proteins have a similar structure. They have a passageway through the protein. They create a polar environment through that nonpolar barrier. They might be slightly different shaped, but they will overall have that same structure of the passageway through the protein. All enzymes are going to have a similar structure. They're going to have a binding site called the active site for the reactant in our chemical reaction. They might be a different shape depending on which molecule they bind to, but they do the exact same thing, which is induce a chemical reaction. There's more diversity in proteins. Some membrane proteins actually get the cell into a certain shape. So there are membrane proteins that are called bending proteins, which actually bend the cell membrane to get into a certain shape. Some cells need to be in a certain shape to function, and these are proteins that can actually get them in that shape. Yes. All right, we'll keep going. All right. Proteins can actually be anchored to the membrane through lipids. So some phospholipids are actually modified phospholipids that have a protein attached to them. This protein could perhaps attach the cell to the extracellular matrix or some other region within an organism. But they're just modified head groups of a phospholipid. You can attach a protein to them, and that is another way that a cell can stay anchored to the extracellular matrix. Okay, we're going to finish off talking about pores and just the overall general structure of membrane proteins. We sort of talked about this reviewing for the first test, but remember, membrane proteins have to have a special arrangement of polar versus non-polar amino acids. Namely, the part of the membrane that's polar, the protein needs to have polar amino acids. And then through the middle of the protein, where it's in the non-polar environment, those amino acids need to be non-polar. And then, of course, the last polar environment, you need to have polar amino acids. This is the only way that proteins can stay embedded within the membrane. If there's a mutation that changes a non-polar amino acid to a polar amino acid, that means that that protein can't stay embedded in the membrane anymore, because that polar region will be trying to form hydrogen bonds with water and would just pull that membrane protein completely out of the membrane. So it's very important that membrane proteins have this unique arrangement of polar versus non-polar amino acids so that they can stay embedded in the membrane. The regions that are on the lipid bilayer, those must be polar, as I just mentioned. And then what spans the actual membrane in that non-polar region, those must be non-polar amino acids. There are a few different ways to do this. So as you can see on this diagram, the red parts of the protein are polar amino acids, and then the blue parts of the protein are non-polar amino acids. For most membrane proteins, their non-polar region is actually composed of alpha helixes. So remember the alpha helix when we talked about secondary structures in protein folding. The alpha helix is the most common transmembrane domain within membrane proteins. But what's essentially happening is it's creating a polar environment through the non-polar environment so that polar molecules can actually get through. Lastly, and this is a short lecture, but I'm recording it so we stay on track, we're going to talk about pores. So pores are the way that water can move through the cell membrane. Water is an extremely polar molecule, as you know, and so water can't just get through the cell membrane on its own. It has to be taken through or have a passageway through. That's where these pores come in. The pores are made of beta sheets. I'll show you on this slide here. Let me zoom in. On this slide, you can see here are the beta pleated sheets, and they form the outside here. So these amino acids are nonpolar, but within the pore, like through the interior of it, those are made of polar amino acids. So it's a polar environment through this nonpolar barrier. And then water can form hydrogen bonds with all of those polar amino acids on the inside, and then water can get through the cell membrane without having to cross the nonpolar barrier on its own. They have to create a polar environment through the nonpolar barrier for water to actually get through. Okay, that is where I wanted to stop, at least for today's recorded lecture. When I see you guys on the next class period, we will discuss passive transport, active transport, facilitated diffusion, and also osmosis. And then we'll be ready for our osmosis lab on lab day. All right, guys, thanks for sticking with me through this.